Hi everyone, welcome to Hari's Success Point. Today in our video, we will discuss about the endomembrane system. That means the membrane system that is present inside the cell. Endomembrane system is nothing but it is a group of organelles which look similar in their structure and whose functions are coordinated. So the endomembrane system includes cell organelles like endoplasmic reticulum, Golgi apparatus, lysosomes and vacuoles. Other cell organelles like mitochondria, chloroplast and other certain cell organelles are not included in endomembrane system because their functions are not coordinated like these four cell organelles. So first of all we will see endoplasmic reticulum. Endoplasmic reticulum is a vast network of membrane-bound branching and interconnecting tubules, vesicles and flattened sacs irregularly distributed in the cytoplasmic matrix. So, they are not arranged regularly but they are distributed irregularly in the cytoplasmic matrix. The first investigations about endoplasmic reticulum were made by Garnier in 1897. It was later on termed as ergastoplasm. So the first name given to endoplasmic reticulum was ergastoplasm. In 1952, it was termed endoplasmic reticulum by Potter and Coleman. The electron micrographic studies of the endoplasmic reticulum revealed the presence of a network of tiny tubular structures scattered in the cytoplasm. So, endoplasmic reticulum divides the intracellular space into two. So, they are luminal and extraluminal. Luminal means the space inside the endoplasmic reticulum and extraluminal means the space outside the endoplasmic reticulum. That means the cytoplasm itself. There are two different kinds of endoplasmic reticulum, namely granular or rough surfaced endoplasmic reticulum and agranular or smooth surfaced endoplasmic reticulum. In short, we call them as RER and SER, rough endoplasmic reticulum and smooth endoplasmic reticulum. The endoplasmic reticulum bearing ribosomes on their surface is called rough endoplasmic reticulum. It is the major location for the synthesis of secretory or export proteins or the proteins that are to be exported to other sites either within the cell or outside the cell. Then smooth endoplasmic reticulum has no ribosomes attached on it. Smooth endoplasmic reticulum is mainly concerned with the synthesis of non-protein substances, mainly lipids. So, it predominantly occurs in cells which actively engage in the synthesis of lipids. For example, adipose cells, interstitial cells of testis, adrenocortical cells. In all these cells, smooth endoplasmic reticulum is prominent. The next component of endomembrane system is Golgi apparatus. Golgi apparatus or Golgi bodies are group of membrane bound spaces and are many in number in secretory cells. Golgi bodies were first discovered and described by Camillo Golgi in 1898. Golgi bodies are essentially membrane bound space. The flat disc-shaped parallel sacs of the Golgi apparatus are called cisternae. The cisternae are arranged near the nucleus. It has two regions, the convex outer face known as forming or cis phase and a concave inner phase known as maturing phase or trans phase. The cis and trans phases are interconnected through a network of membrane. The principal function of Golgi apparatus is the packing of materials that are to be transported to various sites either inside the cell or outside the cell. Materials that are formed in the endoplasmic reticulum are packaged into small vesicles which fuse with the forming phase of the Golgi apparatus and then they move towards the maturing phase. So, during this movement, certain modifications occur in these materials. They also helps in the modification of proteins synthesized by the endoplasmic reticulum. Then the next component is lysosome. Lysosomes are the vesicular, membrane-bound and enzyme-filled cell organelles concerned with intracellular or extracellular digestion. They were isolated and named lysosomes by Christian de Duve in 1995. The name lysosome signifies their digestive function. Lysosome is a fluid-filled bag formed by the packaging process in the Golgi apparatus. The matrix of lysosome contains highly powerful hydrolytic enzymes. Hydrolytic enzymes means the enzymes that are capable of hydrolysis. They include lipases, proteases, carbohydrases, etc. and they are active at the acidic pH of 5. These enzymes are capable of digesting lipids, proteins, carbohydrates and nucleic acids. The lysosomes are important during the time of apoptosis of a cell. Apoptosis also means cell death. Towards the end of the life of a cell, different cell organelles degenerate with the help of lysosomes. Then the next one in the endomembrane system is vacuole. Vacuoles are membrane bound space found in the cytoplasm. They are small and many in animal cell, while it is very large and single in plant cell. A vacuole contains water sap, secretory product and other materials that are not useful for the cell. The vacuole is formed of a single membrane called the tonoplast. In plant cell, the vacuole accounts for 90% of the cell volume and it provides a definite structure to the cell. It also performs the transportation of ions and other materials. In unicellular organisms like amoeba, contractile vacuoles are important for excretion and food vacuoles are also important for engulfing the food items. Especially in certain protists like Paramecium, contractile vacuole perform the function of osmoregulation also. That means maintaining an ionic gradient within the cytoplasm of the cell. So, these are the major organelles that are the part of the endomembrane system.